No, you were going to tell me about the excitement.
10 years ago.
10 years ago.
10 years ago. So 10 years ago, almost to today, 26th of June, there was this big announcement by Tony Blair, who was then the Prime Minister, and Bill Clinton, who was then President in the US, about the sequencing of the human genome. And that was the culmination, really, of 10 years, more than 10 years' effort to try and work out the DNA sequence of the human genome. And that's it's been called various things. It's called the Book of Life. It's the plans for a human being. It's whatever. I mean, there's all kinds of things that people call it. But essentially, it's a very, very long molecule that's made up of 4 different kinds of units, bases they're called, and they have 4 different letters, and so you can express it as just a very, very long word. And that word is 3 billion letters long for human. So 3 billion letters. That's kind of a big number, but it's hard to visualize, it's hard to think about. And one way to think about it is if you were to print it out on A4 sheets of paper, like we've got here, just in a normal typewriter font like you'd use for writing a letter, and then you stacked those pieces of paper up one above the other, then the stack of paper would be as tall as a 5-story building. So it gives you an idea of just how much, it's not War and Peace. It's a lot bigger than War and Peace as a book, or the complete works of Shakespeare, or any of these things. And so that's the genome of one organism. That's the genome of one human being. So that was a huge excitement we had, in the year 2000, and it's 10 years on from that now, and the excitement hasn't stopped. We've had massive expansion in science as a result of that first genome. But what's happened in the last 3 years is the technology that we use for sequencing DNA has gone through another revolution. So as I say, it took 10 years to get the first genome, and I think something like $3 billion. Now, the current technology that's come out in the last few years, you can sequence an entire human genome, not in 10 years, but in 3 days, and for a fraction of the cost, thousands of times less money. So we're talking now within probably the next year, you can sequence a whole human genome for maybe $1,000. So we've gone from $3 billion to $1,000, and that cost is going to just keep falling. So already we know the complete sequence, the complete code for not one human, but now I think something like 15 complete human genomes have been sequenced, probably more. And there are projects in progress now. There's one called the 1,000 Genomes Project. So the plan with that is, as it suggests, to sequence 1,000 humans, and that's well underway. There's more projects to look at disease. So the reason you want to know about the genomes is because we're all different as individuals. And by studying differences between individuals, you can start to understand why maybe one person's more susceptible to a disease than another. So you can think about targeting therapies, you can think about targeting particular drugs to treat one person more successfully than if you just have a drug to try and treat everybody. Because we all know that drugs, in one person it will be a success, you'll get better. Somebody else, it won't make any difference at all. And maybe for a small proportion of people, they'll get a very bad side effect. So if you can, and some of that comes out of your genetic makeup, we're different. So if you can sequence the complete genome of individuals, then you can understand that genetic makeup, and you can hopefully target the drugs. So what all this is doing is generating huge amounts of information. We've got the 1,000 Genomes Project. Of course, this is just human. We don't live on this planet alone. We've got plants, a huge number of different animal species. And by studying different animals and plants, we can understand differences and similarities to humans. And it's possible to do experiments in animals and plants that you can't do in humans. It's not possible for ethical or other reasons to do them. So experiments in model organisms like plants and other animals is very important for understanding what happens in humans. So we need to know the genomes of many different species, and we already do know this. We also know them for pathogens, for bacteria, viruses, parasites that are diseases of humans, plants, livestock, animals, and so on. And by understanding the sequences of these pathogens, you can understand how they're similar or different from human. And it's the differences that you want to target, perhaps with a drug. You want a drug molecule that's going to target some feature that's special to the pathogen. It just only happens in that bacterium, or only happens in that virus, or it's something very special to that that will just destroy that bacterium, but won't hurt the human host. So that's what you're aiming for. It's a magic bullet in drug discovery. So all of these things can stem from knowing this basic information about the human genome sequence and the genomes of many species. How many species? Well, there's millions of species on Earth. We don't know exactly how many. And in terms of the projects that are currently underway, there's a project called the 10,000 Genomes Project. We've got the 1,000 Genomes Project for humans, and there's now a project just trying to start 10,000 Genomes Project. Again, this has come about because the technology now has made it cheap enough to think about actually sequencing 10,000 genomes of many different animal species. So 10,000 genomes, that's an awful lot of DNA sequences. So we've got, at the bottom of this, we've got lots and lots of sequences. We've got lots of these stacks of paper, which are as tall as a 5-story building. In fact, some genomes are much bigger than the human. If you look at some plant genomes, they're 5 or 10 times bigger than the human genome, which is surprising but true. So you've got very, very big stacks of paper. You can't do it, obviously, with paper, so it has to be done with computers. And that's really where my research comes in, and my expertise in computational biology and bioinformatics. It's called a field called Bioinformatics. And so what we do is we take these data that are coming out of these massive projects to sequence DNA and try to, well, we don't try, we succeed in making sense of it by building effective databases and also computational tools to mine, to search through the data, to look for similarities, to understand what the differences and the similarities are between different genomes mean in terms of some underlying biology, some experimental understanding. And one of the big strengths here in Dundee, and particularly within the College of Life Sciences, is the collaborative nature of the research that goes on here. So we sit doing computational analysis, but the biggest power comes out when we can come up with predictions or suggestions which some of our colleagues who are world experts in different aspects of the laboratory-based science can then test those predictions in the lab, or it gives them new ideas to think about how they can take their science forward. And vice versa, they come up with problems from their research which our computational experience can help to interpret and understand more effectively than they can. So this is an extremely powerful kind of interaction that we have here. That's just sequence data. This is just the tip of the iceberg. It's a huge iceberg already, but it's just the tip. Because for every genome and every organism, every individual you look at, you've not just got one thing you're looking at. You're not looking at one cell or one organ or one tissue. You're looking at many different aspects there, and you're looking to see I'm running out of steam here. Let's think. I've lost my thread. I'll come back. Let's come back.
That's absolutely fine.
And for example, one of the things that's very important to study is which genes are switched on and off at particular times in which cells. And this is something that's studied very heavily in this complex in the CLS. So, for example, in development, if you go from an embryo, as an embryo is developing, you have to turn on the embryo. Within the embryo, certain genes have to be turned on at precise time points for the embryo to develop normally. And if those genes don't get turned on, you get abnormal development. So these are very important processes to understand. The other thing that's very important is understanding what's the effect on these genes being turned on and off when you treat with a drug, for example. So you may the kind of experiment that people often do is to have a tissue or cells or a bacterium and look at all which genes are turned on in a normal state, give it a drug, and then see what happens, see which genes change. And that can help understand then how that drug is acting. Because for a lot of drugs that are in everyday use, we don't really understand the details of how they work at the level of which genes are turned on. We know they work and they have an effect at treating a particular condition, but you don't know the molecular details. For some drugs you do know a lot of detail, but for many you don't. So these kinds of techniques can help to understand those processes. And this is yet another aspect of the revolution that's happened since 2000 with the genome sequence, is that once you have the whole genome sequence, you can start looking not just at individual genes to try and study those and what they do, but you look at everything at once. So there are technologies called transcript analysis, which is where you're looking at all the genes transcribed. There's proteomics, which is looking at all the proteins, which are the next level down. There's metabolomics, which is looking at all the small chemical molecules that are produced that aren't proteins and aren't DNA. And each of these different techniques itself produces masses of data. And this is where imaging can come in as well, because by tagging proteins or tagging parts of the DNA with a particular molecule that lights up when you shine a laser on it, you can track where that molecule is in a cell under a microscope. So this produces image data. So you look at the movement of particular molecules in the cell, where they're going. You can't really say what they're doing, but you can say where they're going and what they're interacting with, what other molecules they're interacting with. And you can visualize that, not just, you can visualize that over time,